I think with this one, because I'm not sure I don't know that we've really done right, wrong stuff with this yet. I want to make sure you're okay. So I'm going to put the answers up. Okay. I'm just going to let you check your own, make sure you know what you're doing, make sure you're doing transcription and translation correctly. This one, I think mostly you kind of knew if you were on the right track, because I think they all start with methionine and end with stop at the beginning, and then that changes. But here it is, just to kind of lay it out for you. It's small. Sorry, I wonder if I can put it up one size at least. Any questions, issues? But understand why they missed anything I missed? All right, so let's finish this sucker up. Let's just got to talk a little bit about epigenetics and gene regulation, and then we will be ready. Well, not quite ready, but that'll be all the notes. Your test is for Thursday. Okay, so that's the last day of this week. I think it'll almost certainly be a test that goes on fourth quarter. Raise your hand if you prefer that to me trying to knock it out and get it on third quarter. Raise your hand if you prefer it to be on third quarter. Interesting. So most of you, not a big preference. All right, cool. I'll do it the way it works out. Okay, let's see. Epigenetics. This is about that 97% of DNA we mentioned on Friday. That 97% that doesn't code for proteins. We used to think it was just junk. It is not. It turns out to do stuff. You don't need to know a lot here, because scientists don't know a lot here. What we are finding is that your epigenome, that is the huge regions of DNA that aren't genes, that we used to think were just structural or leftovers from evolution. In other words, stuff you don't use anymore, but maybe your ancestors did. There's some of that. But what we are finding is that the epigenome is important in regulating how genes work. And that, contrary to what we've always believed, your environment can mess with your epigenome in certain ways. Specifically, what we've discovered is that DNA gets methylated by certain environmental factors. Do you remember what a methyl group is? Remember what meth, the prefix, is for in organic chemistry?
Was it 2 oxygen?
Mm-mm.
1 carbon?
1 carbon, excellent. 1 carbon is meth, right? So methane was just a carbon with 4 hydrogens bonded to it. So methylation just means sticking carbons onto the outside of the DNA. And it turns out that that happens to DNA in certain environmental situations. And that DNA that gets highly methylated is less likely to get used. So if portions of the epigenome near certain genes gets a whole bunch of carbon stuck onto them, those genes aren't as likely to turn on. When you learned about evolution, whatever year it was it 7th grade? Anyway, did you learn about Lamarck? Lamarck is basically famous for being wrong. Lamarck was around the time of Darwin. Lamarck's ideas were that, for example, a giraffe was an animal that during its life had to stretch a bunch to reach the high leaves. And in doing that, it stretched its neck out physically during its lifetime. And that stretchiness got passed on to its offspring. That a snake was just a lizard that hadn't used its legs enough. And so the lizard's legs withered up, and that withering that happened during its lifetime got passed on to its offspring, and gradually we got a snake. Not a crazy idea, but we know it doesn't work like that now. We know that evolutionary change happens between the generations, right? It was that the giraffes that were already had longer necks genetically did better than the ones that had shorter necks, passed on the longness of their necks to their offspring, right? That the lizards that had shorter legs must have had an advantage, and so that's why and snakes and lizards are kind of a different story anyway, but like a skink or whatever. Wrong, right? It's a famous example of, hey, it doesn't work like that, it works like this. Except epigenetics is messing with that a little bit. What we're finding out in epigenetics is, though we used to think that characteristics you acquire during your life can never be passed on, that's still pretty much true. You know, you can lift weights as much as you want, get as big as you want. Your kids aren't going to be stronger because of that. But people also tend to kind of think of it as a free pass. Like, I can do whatever I want, and I'm not endangering my children because it's not changing my DNA. We are finding that certain things that people do or go through in their lives do create changes that get passed on. And it's about the epigenome. I'm not going to give you a lot more detail than that, although this is, I'm sorry, this last paragraph. It's always the things that we don't know everything about that we have to write more, just because I can't give you the simple way it works. But what we're finding is that some things that you do during life or that you go through during life cause changes in your kids, even in your grandkids. The first study that was really conclusive about this had to do with Dutchtown. Scandinavians, I don't know why. They must just keep really good genetic records. But a Dutchtown basically went through an entire year of starvation during World War II. And what they found was that period of starvation in those Dutch townspeople, the ones who had it worst, their grandchildren, literally like not one generation, but two generations, to their grandchildren, they found increased risk factors for certain genetic disorders. So like that thing that happened to those people in their lives somehow caused problems for their grandchildren. That's not how it's supposed to work, right? So opened up a whole field of epigenetics where, you know, we're looking much more closely now at the DNA that doesn't code for proteins. It's not nothing. Questions? I know it's kind of vague, but that's where we are. It's a kind of gene regulation that we don't completely understand. So now I want to talk about a couple of very simple kinds of gene regulation that we do understand. Everybody good? You guys ready? Okay. First of all, we know that genes tend to be regulated in groups called operons. And I'm just going to teach you about one very simple bacterial operon. Bacteria are nice because they have simple systems that are easy to understand. It's called the lac operon. Turns out bacteria, specifically the ones that make milk go bad, the ones that make milk into yogurt, separate from that, there are actually quite a few bacteria that like to break down lactose, which is milk sugar, if you remember, which is glucose and galactose. It's a disaccharide. Well, in order to get energy from lactose, you got to split it into the monosaccharides, right? Into the glucose and the galactose. So prokaryotes have a group of 3 genes called the lac operon that they use to do that. And in its normal state, the place where the RNA polymerase, remember RNA polymerase does transcription, place where that's supposed to bind is normally blocked by what we call a repressor protein. Just a protein stuck to the DNA, keeping RNA polymerase from binding, from transcribing, from the proteins that break lactose down from being made. And so let's say this is, gene 1, gene 2, gene 3. Got this spot here. You've got the repressor protein. Blocking RNA polymerase from binding there. Well, turns out the repressor protein has a little nicely shaped notch in it. Guess what fits in that notch? That changes its shape, knocks it off the DNA, and allows RNA polymerase to start transcribing. What would make sense that would fit in that notch? Lactose itself. The presence of lactose. Lactose fits in that spot. Ooh, what's happening there? Lactose fits in that spot, makes the repressor fall off because it changes its shape, and polymerase can bind and transcribe and translate those 3 genes, which allows the cell to break the lactose down. This is bacteria we're talking about. So it runs those 3 genes until the lactose is gone. As soon as the lactose is gone, the lactoses that are in these slots of the repressor proteins get broken down. The repressor proteins glom right back onto the DNA and block the polymerases, and the lac operon shuts down. Very simple, elegant gene regulation, right? So lactose is the thing that binds the repressor and knocks it off. Questions about the lac operon? It's about the simplest gene regulation thing I know. And that's really the only specific example. The rest is kind of general. Eukaryotic gene regulation is more complicated. Of course, it's more complicated cells. Here are some things that happen in eukaryotes. I mentioned before, and it was in the video, the ta-ta box. Sounds funny, but it's just that it's thymine, adenine, repeating. That exists upstream of many genes and is a place that helps the RNA polymerase move into position. So blocking the ta-ta box can regulate, can block a gene. Opening it up can turn a gene on. One of the things about DNA is that it undergoes various levels of coiling, right? Like it packs way up into chromosomes when a cell's dividing. That's obviously not when it's being transcribed. But at other times, often it is coiled up and not being used. So there are things called transcription factors that are special proteins that can turn on genes by opening that coiling up, by unpacking packed up chromatin. or by binding to the top box and allowing the RNA polymerase to bind. Finally, we are finding more and more things that RNA does besides, outside the mRNA, rRNA, tRNA jobs. And one of those things is that if you've already transcribed a gene, right, you've got the mRNA transcripts out there being translated into protein, and you want to shut it down quick, one way to do that is with what we call RNA interference. You basically make little chunks of RNA that'll bind to the mRNA that's already out in the cytoplasm and keep it from being translated. That is all, folks. That's the chapter. Test Thursday. I have two things for you today. Number one is weird. It's two activities I made up. Both of them are like Sandro level weird. One is, do you remember the thing we did in the cell chapter by the cartoonist who only uses the 1,000 most commonly used words in the English language? Okay, so he also made a text editor where you can type in that and it'll restrict you to only those words. So what I did was try to explain gene expression just using those words. And what you got to do is identify what I mean by each of the bold phrases. Each of them correspond to something on the word bank on the front that is all the parts of the stuff that do gene expression. The other side is an analogy that I made up. It's a dumb analogy. It's a weird story about a car company. But it's told in such a way that there's an analogous part all through this story. Each part that has bold print corresponds to the function of something in gene expression. Analogies are maybe the most powerful way to make sure you understand how a system works. So that plus the 1,000 words thing both share the same word bank. You just got to kind of choose which parts, it's like a big matching thing. Choose which parts work. I will tell you, you will be assessed on how you do on this, but I will take the higher of the two scores because I think they kind of press different buttons. And so some folks do better on one, some folks do better on the other. So as long as you do really well on one of them, you'll get a good score once we go through it. So that's thing one. Thing two is sample essays. Both of those things are due Wednesday. And basically, whatever I post tomorrow isn't going to be a whole lot. This is going to be most of it.